All right, I think we're good. All right, welcome everyone. So welcome to the INC-IEM Neuroengineering Seminar Series. And we're very delighted to have with us today Sandro Carrara, who comes from EPFL. So a few words here. I won't go over the long list of awards and all this recognitions. It's wonderful seeing and knowing Sandro for many years as
good friends.
Good friends,
exactly.
Absolutely.
So it will be unfair to present me the right way, right?
Exactly. Yeah,
because we are friends.
Sure. No embarrassment here, but so Sandro has made really great contributions in the field of sensing, circuits and systems. In fact, he's part of many societies of IEEE, right? And having grown up in Italy, then have been in Switzerland for quite a while. At the EPFL has done really amazing work. So the really exciting part today is going to tell us about metabolic sensing, human metabolic sensing, remotely, and with the Nano, just everything from Nano to
everything we need.
Because of Nano, yes. Yeah.
Thank you, Gert. First of all, thank you, Gert, for having invited me for this seminar. Thank you all for being with us today. As Gert said, we are going to present bio-nano and electronics interfaces with aim of remote monitoring of human metabolism. So on the way, just to understand what we would like to speak about, we can just refer to what someone called distributed diagnostics, the fact that you can, for example, use electrochemical sensors as kind of plug-in or little interfaces to, for example, transform our mobile phone in a glucometer. This is an interesting case. If I'm not wrong, it's an American company that is introduced this device years ago. This is the latest novelty by Abbott. You have seen, I'm sure, somewhere during summer carrying this white button. Looks like a wearable. Actually, is not really a wearable because it has microneedle that break the skin. It contains, again, electrochemical sensor and electronic front end. And then we have an electronic user interface. So just to have a little bit of look in perspective, this is a quite old estimation that was, foreseen by 2025, kind of $7 billion equivalent of revenue with 3 billion wearable devices. And actually, more recent seems to have confirmed 2022, 61 billion US dollar, even more than expected. So what we can see in this figure is that, continue with this growth, we will probably, for a say, we will move from 3 billion in 25 to 26 billion by 2030 of internet connected distributed devices. And in these huge, enormous, to me, skyrocket figures, half are related to consumer electronics and 1/4 to health. So 2/3 will be for our personal electronics related or not with our health by 2030. If we remain with this 1/3 for health, the plan is, of course, to tackle this quite old idea because Mr. Deman gave me this, offered me this slide 15 years ago. So it's already a while that we are targeting this, vision at the end of the tunnel to have, humans fulfilled by hidden technology, by hidden sensors, in order to be brought directly in contact with the whatever medical system stack or department by through internet connection. Well, if you see the mentioned sensors, you can immediately recognize that some of them are already there. ECG, EEG, hearing, they are, state of the art in the market now. But for some of them, toxin, DNA, protein were still research need. We have not only no product in the market, but we have not exactly well-established technology to succeed on that. So what we need to do to succeed on that? To succeed on that, we need for sure to integrate in our sensor biomolecule, typically to assure specificity or selectivity, probably better to say and to speak about selectivity. Often we need to integrate nanostructure in order to assure sensitivity, enough sensitivity. I will show you cases about that. And then, of course, if we want autonomous systems, so a mobile phone that played the role of a diagnostic tool, we need, autonomy. That means we need high-level function. That means we need close electronics. Nowadays, we need also computations on board more often. Someone called it edge computation. Someone started calling, in some cases, on-sensors computing. That means we need even more electronics on board. So on this slide, usually I'm happy to highlight three keywords, typically: the bio, the nano, and the CMOS, which drove me now more than 15 years ago to develop this concept that we need to co-design the interface between bio, nano, and CMOS. And I have to be honest, I came to this conclusion after having seen several projects failing, because we were, for example, designing only the electronics and then looking for the right biosensor to couple, or vice versa, designing the biosensor and then looking for electronic to couple, and always fail on that. I made a career on this. I wrote a book. This year I'm out with the second edition. So if you want to jump in more, please feel free to. Thank you. Feel free to go there and to have a look of the book. Today I will try to give you some needs, some starting needs about what does it mean to co-design bio, nano, and CMOS. So let us start first with bio. So if I want to detect a series of molecules, and in principle, as we joked during lunch with Gert, in principle, every molecule might be detected, but devil is in details. So we can detect ions, drugs, glucose, lactate, cholesterol, whatever, endogenous or exogenous metabolite. Please don't forget that the glucose is half of the worldwide market. That's the reason many people work on glucose, but there are plenty of other diseases than diabetes that require close monitoring of typically markers of the disease, endogenous marker. And in the area of precision medicine, we would need to measure the drugs, the therapeutic, in order to adjust the performance of the therapy on the exact metabolism of that single patient, right? So the question now is that how to measure all of this. Case of the glucose, I don't discuss because it's too known. Lactate and cholesterol may be detected in a similar way. Let us consider a bit more strange case, the case of the drugs. So what happens if I have a drug, if I take a pill, and what happens in my body? Typically, millions of years of evolution of mammalians have developed another class of enzymes in our body, not in body of any mammalian, that is the class of the cytochrome P450. There are more than 3,000 isoforms of the same kind of protein, and this protein has been developed exactly to treat exogenous compounds. So how they work? Let us consider one case. The cytochrome P450 2B4, one of these 3,000, catalyze the bensphetamine, which is a compound for the mind. How it works? The protein typically manages the bensphetamine by consuming oxygen, and with the help of a coenzyme, the NADPH, and protons, and typically oxidize the drug. Why this? Because the oxidized form is more soluble, then faster to be secreted. Never forget that when we take a pill, our body immediately thinks there is an alien in the body, and it's not immediately to bring out. The phenomenon is called clearance, and the clearance is the reason why the doctor, when you bring your kids to the school, to the doctor for a flu, is say, okay, give him 1 antibiotics every 8 hours, because in average, it is known that every 8 hours, an antibiotic is cleared by the body. But this is an average. And believe me, I don't have statistics to show you in this presentation, but the standard deviation patients may reach 200% of the average. So to say 8 hours means nothing with respect to your baby or with respect to yourself. But, and it's clear to you now why, because each of us has his own amount of cytochrome, each of us has his own wild type form of cytochrome, more or less perform. So this is the pathway in our body for our aim of technology. If we look closer at the chemical reaction, we discover that it's a redox reaction. So protons and the NADPH typically provide electrons. But if we are in business with electrical engineering, electrons is our daily bread, right? So if I provide my electrons to the chemical reaction, I'm in business because I can just measure a current and quantify how much drugs has been transformed by the enzyme. So with this in mind, I can develop, of course, biosensor based on cytochrome P450. Another case is the ions sensing. How I can do it? I can use so-called selective membranes. So I can produce artificial membranes that are selective for the potassium in this cartoon, or for sodium, or for whatever other ion, and then typically register open circuit potential at the electrode corresponding to change of the ion. The main drawback is that, as is shown by the cartoon, typically the measured open circuit potential is not enough stable. You will see data in a while. But out of that, this is possible. I measure a potential in this case, not current, and I get a detection of an ion. Okay, so what's about Nano? About Nano, I have a good example to show you. This is a cut from literature. It's a detection of another therapeutic compound called verapamil, which is an antihypertensive drug, as detected by another isophore of the same cytochrome P450 I showed with you right before, 3A4. So all the different isophores are called by two numbers and one name. Apparently, we do a bright detection, fairly linear in the millimolar range. But now, if we want to develop a real technology, we need to compare this calibration curve with the concentration we need to detect in the human blood, in patient's blood. And as you see, the range is below 0.3 micromolar. No way to have a real technology out of this in principle detection. Devil is in detail. Luckily, in some cases, for example, multi-wallet carbon nanotube may help us a lot. This is another example from our lab, cyclophosphamide, very well-known anti-cancer compound, detected by the same enzyme, the 3A4 of the verapamil. Again, if I just drop the enzyme in my electrode, I get a fairly good linear calibration curve, but the calibration curve starts exactly where the therapeutic range ends. So again, I'm out of any business because my sensor is blind where I need to look for the drug. Instead, it was sufficient to just add carbon nanotube, and we had succeeded in fitting very well with the therapeutic range used in patients' blood. So you see, the integration of a nanomaterial bring us back in business in using this kind of enzymatic amperometric sensor. How we can integrate carbon nanotube? We have many different options. I just present you one to you, just to take it shorter today. So we can, for example, define our multi-panel platform, transfer by electrochemical deposition the precursor film, and then typically flux carbon-based gases to grow nanostructure on top. This is typically done at high temperature. Have a look at the numbers, are incompatible with CMOS design. But luckily for a work of a bright PhD student, I had the pleasure to work in past. Now she is assistant professor in Catholic University of Leuven. She succeeded in getting fairly good yielding below 450. So entering compatibility with CMOS design, CMOS circuits. How to obtain different kind of structure? Well, Irene discovered that we can play with different quality in the precursor film, in particular non-compact precursor film, or compact, or so rare that cluster in nanoparticles. So by starting from this kind of amount of electrodeposited precursor film, she succeeded in getting the typical carbon nanotubes, more fatty, more longer, depending on the size of the particle she had as a precursor film, or in very compact precursor film, she obtained kind of roses that again nanostructure the electrode with typically a graphene walls. Play on that. This is just one example from our paper. You can see that optical microscopy show that we are increasing the number of nanomaterial on our working electrode, and we are going up, for example, with the detection of the glucose. Year later, again with Irene, we started playing with nano gold and nano platinum, which is my way simplest than carbon allotropes, because in this case, we can just use electrodeposition by divalent or tetravalent salts of platinum or of gold and obtain nanoparticles or a kind of nanowalls. And in this scanning electron microscopy graph image, you see both nanoballs and successive deposition of walls. So we can obtain again nanostructuration of the electrode just by electrochemical deposition. This is much way better for our electronics because we can do it directly on CMOS die without any heating. It's just sufficient to add the function in our CMOS architecture to produce by electrochemical deposition the nanoplatinum. And you see this is a work done in collaboration with Imperial College, another former student of mine. Now she is lecturer in UCL. We deposit nano balls, and then on top of the nano balls, we have deposited walls of the platinum directly on top of the exposed metal layers from the die. So easiest for CMOS integration. With this kind of nanomaterial, nanoplatinum, we got great advantage in ion sensing as well. I mentioned you the fact that there is, there are variations, non-stable potential when we try to measure with respect to whatever ion. This is typically due, literature said, by a presence of a water layer impossible to avoid below these selective membranes. But it seems that this layer is gone if we play with nanostructured electrode. And here you have the proof. You see, non-structured platinum electrode, fairly large drift in few hours, both nanoplatinum and nanosphere, same membrane, same set of experiment, dramatically stable open circuit potential. So mission, if you like, accomplished. Is not to the sake of sensitivity, it's to the sake of stability of the measurements, but in any case, nanomaterial give us an extra advantage in developing our sensors. What's about the electronics? So for the electronics, again, there is a word to discuss. I said I wrote a book. There are 5 chapters dedicated to the electronics. I will just show you one case, the typical amperometric detection. I mentioned you enzymatic biosensors. They are typically done by exploiting one working electrode, one reference, and one counter. And we need first to stabilize the potential between the counter and the reference. And we might do it with a control amplifier organized in this manner. And if we do that, we can rely on the typical characteristic of the operational amplifier that does not require any current in its inputs because it's an enormous impedance, which means we are providing a potential at the reference without providing any current that make the reference stable in time. And by definition, by international convention, the reference in electrochemistry. The only thing we need to take into account are the typical convention in electrochemistry that usually marks the potential at the working with respect to the reference. Instead, we apply a potential to the reference. That way, our bias is to the reference. That means we need to take into account the reverse of the sign with respect to the Nernstian potential we need to apply. Back to the case of the cytochrome P450, the Nernst potential of the peak is around 380 negative millivolts. We need to apply there plus 380 to reach a current that is related to the Faradaic process due to the cytochrome P450. What's about the current? Once I have the right Faradaic process ongoing because I'm controlling the potential, then I need to acquire a current that, as per the example I gave you, will be proportional to the bensphetamine. Well, in this case, I can just use a transimpedance amplifier I will get a without potential voltage that will be proportional to the feedback current, which is reversed with respect to the Faraday current collected at the working electrode, but ultimately will be proportional to the concentration of the species that are going through my redox reaction in the electrochemical cell. This is just a transimpedance amplifier, so a guy that converts a current to a voltage, as you know, probably better than me. But there is a smarter way to detect the concentrations in an electrochemical cell or sensor. I love this idea by Nano and Harris, published in 2006, where they proposed to manage the electrochemical sensor, as usual, with the control amplifier done by operational amplifier to control the potential at the reference. But then the smart idea is how to read the concentration. They use typically a capacitor fed by VDD in order to charge a capacitor, and then control this charge by a comparator, which eventually switch when a threshold is reached. This switch is used to switch off the feeding transistor, the transistor that connect this capacitor to the VDD, and then let the capacitor to discharge by through the electrochemical sensor or the electrochemical cell. What does it mean? Means that the timing of the discharging of the capacitor will be driven by the Faraday process in my sensor. So as much chemical I have, as much Faraday current I have, as faster will be the discharge of the capacitor. So at the end of the day, what they obtained is a current to frequency converter, no need anymore to an ADC, just a digital output, and the frequency at which this output change is proportional to my concentration. This is very good, especially if we want implantable device with minimum transmitter. Just showing you a case. You see our chip inside the peritoneal region of the poor mice. Here is closed, and then we put the mice in a cage to measure several parameters: pH, temperature, ATP, glucose, and a drug. For example, we did with paracetamol. Why it's an advantage? Because as you see here, if you pay attention, there is a column in this image. It was an inductive link to transmit power. So that means we are providing power to the system by an inductive link. You know, by basic courses in electronics, that the power transfer is effective if the two networks are matched. So we need a perfect match between the transmitting network and the receiving one. But if we use, for example, multiplexing and a current to frequency converter by the idea of Narolas and Ari, we can use the output of this converter to command a switch that bring in and out the full matching of the two networks. So we will be, roughly speaking, alpha period matching, alpha period not matching. What does it mean? It means that we will have a shock back of the non-matching period. So we will measure from the external power manager a matching, non-matching situation, which frequency is proportional to what we are measuring. And thanks to the multiplexing, it will be proportional to the glucose, to the ATP, to the temperature, or to the bensphetamine if we are using a bensphetamine sensor. So as I mentioned you, the cytochrome might be used to measure more than one drug. I showed you cyclophosphamide, but also verapamil. Well, the really bad news, this cytochrome is really poorly selective. So for example, the 2C9 work also in naproxen and fulbiprofen. And even worse than that, if you pay attention on this data, we have shown that keeping the concentration of fulbiprofen fixed at 200 micromolar, this is the peak of fulbiprofen. If we increase the naproxen, fulbiprofen and naproxen are two very well-known anti-inflammatory compounds, are typically used in the cream for the muscle that you have muscle pain. Have a look. I'm going up with the naproxen correctly. The peak of naproxen goes up, I have more Faraday current, but also the fulbiprofen goes up. But the concentration of fulbiprofen doesn't change it. What does it mean? It means that the presence of the naproxen activated better the enzyme to work on ibuprofen as well. And I have no other slide in this presentation, but if you're interested, I can show you. I have even the opposite case. So increased concentration of one compound give less current from the same concentration of the other compound. So this is just to mention that cytochrome P450 is a bad boy. And that's the reason I'm so fascinated to work with that. I worked with that over the last 30 years of my life. In this moment, there are big projects running on it where we are trying to use machine learning approaches to read out by this complexity. We want to measure 6 anti-cancer compounds simultaneously, and I try to use, machine learning to have automatic recognition of the peak and, of course, automatic recognition of the mutual interaction of different drugs on the same sensor. Okay, of course, in any case, from the electrical standpoint, for the circuit standpoint, I need to sweep the voltage. I cannot necessarily do typically by charging and discharging a capacitor. That is a typical situation, the typical solution chosen in CMOS design for high frequency. The reason is that we are generating sub-Earth signal. We need to measure to sweep this voltage 10 millivolts per second over a range sometimes of 1 volt. So we are definitely sub-Earth. So in order to do that, we have proposed years ago, a kind of digital synthesis of the signal. You can use typically a word and then an DAC, so a digital to analog converter, in order to obtain typically a voltage ramp, a voltage ramp that is then used to acquire the current at different potential. What's about ion sensing? The electronics to sense ions. Well, I mentioned you that we need first to deposit a selective membranes. If the selective membranes is in iron oxide, we can measure pH as well with exactly the same technique. But the problem is that we need to measure a potential. And this for sure will be not an ideal voltage supply, right? If we attach whatever current, if we attach whatever circuit with the suck current, we will see whatever voltage, not necessarily what is the open circuit voltage. Second problem, we need a reference. They are not absolute voltages, but they are voltages related to what happened in my electrochemical interface. However, if we have amperometric sensor on board, I have already showed you that we have somewhere a reference selector that is already fed by my control amplifier, right? So what I can do is that to use a voltage follower to try to read the potential at the the ion sensor or pH sensor without sucking any current, and then I can compare this voltage with the voltage I'm using to the reference electrode. In this case, for example, this is a experimental data on pH. You see they're fairly linear, working very well. We have done exactly the same for potassium or for other ions like sodium, for example. So I showed you the different idea we are usually using. Okay, everything seems to be fine. So we have nanomaterials, we have biomaterials, and we have MOS material. We can integrate everything together and then go for application. So first of all, just a gallery of some of our glories. You see the years when you have developed these different chips. And then, for example, we have applied some of them to the metabolism monitoring. I just offer you some comparison. We have typically compared the Autolab potential stat, which is considered kind of Ferrari in electrochemistry because it's a very robust and very costly instrument, with similar measurements, same sensor, but with our ICs. So you see the comparison, for example, in glucose detection here, or in lactate detection in chronoamperometry. So we fix the potential, we just follow the current. You see very similar within the measurements error differences. We have done also a comparison direct between sensitivity and limit of detection. Of course, the bad news is that in the limit of detection, we are doing very badly with respect to the Autolab. Instead, the sensitivity is good. The reason is quite obvious, right? So the Autolab is machine bigger like that. You can imagine how many filters are inside our chip. It was the second one. It was 1.5 millimeter by 3 millimeter. So not that much filtering on board. And so that's the reason we are getting worse in terms of limit of detection. By the way, sufficient for the application because the limit of detection is in micromolar. Both lactate and glucose in humans are in the millimolar range. So not a big deal for application to wearable or to implantable. We have also done in voltage scan and cyclic voltammetry. This is the detection of another very well-known anti-cancer compound, the etoposide. And you see again comparison between our electronics and the Autolab. In this case, what matters, as you have seen before, is establish where the peak is. And in fact, we have quite good peak location of the two concentration, 200 micromolar and 400 micromolar of etoposide, both with Autolab and with our ACs. So apparently mission accomplished. So let us now have a look of how we can do remote monitoring with all this stuff. So I will go to show you some applications, some systems that we have developed and successfully tested, almost always successfully tested over the last kind of 15 years in EPFL. We have done portable, we have done wearable, and of course we have done implantable. You have seen the case of the mouse. So this is one of the systems we have developed for intensive care unit. It was a cube, 3 centimeter cube that was hosting on site. You see here the platform, several gold electron, several clusters provide one sensors, and then underneath you see the electronic with our integrated circuitry. We have demonstrated possibility to detect glucose and lactate that are key in intensive care units. Typically, when in case of organ failures, the lactate goes up as a sign that the cells are going to die. The cells are going to die. And we have also simulated organ failure through the measurements of the potassium. So I've made the in vitro experiment. We have led the cells literally to explode, and we have measured a huge wave of the potassium that is again a typical mark of organ failure with the potassium sensor I showed you before. Another good example of our application was in anesthetic monitoring. We have been required years ago to develop a technology to monitor the anesthesia. So as I told you, every exogenous compound is managed differently by us. Can you imagine the difference during the anesthesiology? I can get the same amount of view. I can never sleep, or I can sleep for half the surgery and then wake it up suddenly, or I can sleep forever. So it's very scary. And nowadays, the automation in the injection of the anesthetic components is fairly good, but it's just based on model, on mathematical model. We have no feedback, but some ECG measurements that is again considered indirect. So we had a couple of projects on that, and the idea of both the projects were let us measure at least sometimes in the patient's blood and return a precise feedback to the injection machine. And that's the reason you see all this complexity. So several sensors, because any anesthesiology is done by a cocktail of drug. Need to make the patient sleep, need to make the patients don't feel in pain, otherwise it's wake up, right? If I touch you where you sleeping, you immediately wake up, right? I cannot cut your skin, clear. You need the patients to be relaxed. So the surgeon need to find a relaxed muscle to cut easy. And then they discovered several tens of years ago that people under this treatment could recall the surgeries in nightmares. So they had a hidden, not conscious memory of the surgery. So they had a fourth component to erase the memory of the patients in order to be sure that you will have no, in the 20 years to come, nightmare about his surgery. So that's the reason you need to monitor more than one compound. Typically, every compound has its own sensor. Then once you measure, you need to go back to the modeling before to command the pump. And here you have, just simple proof that we have measured at least 3 components, midazolam, paracetamol, propofol. By the way, this compound is the compound that killed Michael Jackson. That's a proof how toxic is anesthesiology. So how dangerous may be the compounds. And here you see semi-continuous, semi-real-time measurements of paracetamol, choledosol, APAP, and propofol. So just to measure that we had on board the possibility to measure simultaneously the 2 components. Then the latest novelty I've done with the second project is to bring all this technology on the hands of the anesthesiologist. So as what we call it, the smart pen. Here you have in principle needle-shaped electrodes. So once the anesthesiologist wants to calibrate injection, can go to the patients, insert directly in the sensor, perform the measurements of the propofol. We have a machine learning approach on board to read out by a problem that is the fouling of the propofol on the working electrode, and then transmit back the data to the machine that commands the pumps. Of course, we can do it something similar on the sweat. I'm in the kingdom of the Joseph Wang, so I'm here to learn, not necessarily to teach. But you know better than me, because you're a bright professor, that we can measure on the skin. Again, David is in detail. We need to have ontophoretic to extract better the molecule from under the skin. But once we are there, we can, of course, insert electrode in our sensor and we can play around. So flexible electronics for the readout and whatever kind of screen printed electrode to read it on board. We have done with several molecules. I usually like to show that we succeed in measuring the nicotine on the skin. And we have succeeded in the identify and classify light smoker by heavy smoker, who one postdoc and one PhD of the group that ran for half an hour with the patch on board. She was a light smoker, he was an hard smoker. And we succeeded in distinguishing the 2 cases with fairly good calibration of the nicotine in the sweat. So I was very excited about that, because this was an industrial actually founding. That time they were interested to see if it's possible to measure the nicotine in a non-invasive way for to develop a product on that. And we had no idea if it would be even possible to detect nicotine on top of our skin. This experiment demonstrated yes. This is the hard smoker, which data I showed you before. Oops, sorry about that. He was used by another of my PhD student, to make a exercise during which we measure, for example, potassium and sodium. But the lady, Francesca, beyond this research, was even brighter, because she also developed a lithium sensor, a sensor for the lithium, which has been largely required for monitoring bipolar patients. Here, the problem is that you don't know if they take the pill or not when they show the same symptoms. And she was very, very bright in developing enough light and enough performing sensor that may be just kept in a bandage and again read on the skin the amount of lithium in that bipolar disorder patient. Well, as you can imagine here, the problem is that minimize the implant, because with the wearable we have much more space. We can play with electronics, bring in contact with our mobile electronics. But the true challenge is really here, how to do it enough small with all the performance we might need. So outside, I said it's not a problem. We can use, even commercially viable tool. In this case, is a quite old version of the Bluetooth technology. Here is typically a coil for the inductive link. I showed you before. We can transmit power and receive back data. But as I said, the problem is inside. So the baby you have seen in the peritoneal region of the mouse has been built with a remote powering antenna, a coil, couple of chips designed by us in EPFL and built, of course, in Taiwan for the electronics. And then on top of the baby, there was, the sensor, and everything was sandwiches and wire bonded, and everything was in a 1 centimeter square, few millimeter thick. This is the real baby, 4 amperometric sensor, 1 pH meter. You don't see here probably, but there is a temperature meter. pH and temperature are necessary to calibrate the work of the amperometric sensor, current shift, both the cases, peak shift or current increase. And here you see biocompatible packaging, typically done in biocompatible silicone. And then there was a carboxylic membrane on top of the sensor. Small enough, will be ever go inside skin of humans, unless they are threat of the life, because we know that pacemakers are currently used for deep brain stimulation, but these patients cannot stand up without the deep brain stimulation. What's about healthy people? Because we are speaking here about, general monitoring of the human metabolism, also for prevention. And, 10 years ago, when I was asking in the class, no one seems to be prone to get it. I've done last semester, and again, the student, show, no, why I need to receive an under the skin chip if I'm healthy? Well, survey in Europe, 2022, 51% of the interviewed said that they are quite ready to get it, not only for health, but just only to pay. When we go to the shop, we show our mobile phone for twint, or we show our wireless contact a credit card. Why not just showing your arms? And actually, I have to be honest, there was a guy already in Switzerland in 2014 that was in the news because he asked to have a couple of injected passive RFID just to be recognized by his door or by his mobile phone. So if we want to stay minimally invasive, similar to what we are doing with our pet animal, because the RFID that are used in this example are exactly the same that in US and in Europe we implant to our pet animal, typically the dog, in order to be controlled under ID control, but they are passive. If we want to do active, we need to integrate molecular sensor, pH sensor, temperature, enough powerful inductive links, and of course, dedicated IC in size of a needle. And actually, we need to stay very small in terms of size, at least like diameter. We have done years ago with this, baby, exactly in that size. And even more importantly, Mark Phelps from Medtronic showed us already in 2015 Biocast conference in Atlanta that they are producing now an ECG monitor that is just injected by a syringe, one point of surgery. This is a big change because that means no anymore surgery to receive electronics under the skin. But you see a big leader like Medtronic has already entered the market with a similar concept. This is at the moment only for the ECG. At my question, Mark replied that yes, they're at least considered to bring on board the glucometer. So you see, it's coming because big worldwide leader in electronics for health are working on that with injectable syringe injectable electronics for health monitoring. Okay, once you are, in your mobile phone or in your laptop, you can, visualize endogenous and exogenous compounds. Once you are in the tablet or in the mobile phone, you can go through Wi-Fi or internet to whatever you want, including the list of the responsible of the intensive care unit, and you can just, show what is the patient's, what is the parameter that is outranged, and what is the range in order to send alarm to the responsible, especially in case in this moment is not in the room. Well, we have seen application to portable, wearable, implantable. So the question is that now what else? So at the end, just fire all these university professors that study wearable because everything seems to be done. Well, yes, in the sense that wearable now is moving, thanks to all we have done over the last 20 years to industry. And then in order to not be fired by our institution, we need to invent something more. What will happen in 20 years from now, right? Okay. We are bright people. you have probably too much fantasy. And so we have decided to investigate at least the idea of the body dust. What does it mean? It means the idea to have kind of dust, intelligent diagnostic dust, that you can in principle spread in our body and get telemetry by this, just smart dust. I know it looks crazy, it looks crazy, but Gartner, not me, not some, Gartner, already in 2018 said that if we look the hypercurve, considering the emerging technology, smart dust will be there. Okay, smart dust, just to let you focus the keywords and the timing, is a keyword that has been invented in University of California, University of Berkeley, San Francisco.
University of California, Berkeley.
Exactly. So University of California, Berkeley. Thank you. I was thinking University of California, San Diego. Exactly. In any case, the same country. We are here. It has been introduced in early 20s, so 25 years ago, right? Okay. Then the concept of smart dust is what nowadays we call it IoT, the Internet of Things. Because if you read the original paper, Market 99 conference paper from that group in University of California, Berkeley, you will see that is the modern concept of IoT. But in the continuation, again, University of California, Berkeley, in 10 years, 12, 30 years later, they introduced the concept of neural dust. And then we try to consider to extend the concept of the body dust. So what is the idea? The idea is that to have, as I said, a diagnostic dust so small that we can just drink a glass of water, apparently with nothing inside, and then get spread that diagnostic in our body. So of course, the main concept is to have enough small CMOS particle containing all the sensors we need that can go through the digestive system, pass through a mechanism that is called internalization to the bloodstream. And once they are in the bloodstream, for example, self-tracking a tumor mass. And while they are there, they can stick on the tumor and provide telemetry, for example. They can stick selectively in the tumor. It's enough to have, the antibodies specific for the tumor cell. The problem is the size of the particle, because if we want to exploit the internalization from the digestion system to the bloodstream, and at the end of the day, the clearance, typically through biliary way, we need to cope with the same size of the blood cells. We would need to be as well flexible, but starting point at least similar size. So in between 7 and 30 micron. How to do it? Well, not easy, because we would need extremely small sensor, extremely small CMOS architectures, extremely small CMOS architecture for the sensor front end, as well as for the transmission backend. It's quite, fairly complex architecture. Then we will need to see how to power this so tiny baby, and then we will need to prove that it works. So internalization and then clearance phenomena may happen, at least in animal model. So let us try to see where we are now. So extremely small sensor, for example, glucose sensor. Well, already old story. 10 years ago. Great scientists, I'm sorry, we have in Zurich. This time is a point for us. Here, Lehmann and his group published, glucose sensor. This is nanoplatinum, same material I showed you before. And have a look, they succeed in getting glucose sensing down to 10.5 micron for the working electrode. So well done. It seems we can do it. Thanks to the nanostructuration, we can collect enough current, of course, smaller current than if we play with 50 micron, but enough current to, for example, detect the glucose in only 5 micron of working vector. was about electronics, because I need that now to bring everything. Of course, I didn't mention, but Hilleman did on CMOS. You see the chip there, but the chip was bigger. So only the working electrode was 5 micron. Instead, if I want to integrate also the potential stat, I need to bring all the transistors I needed in 10 micron by 10 micron. So already in 2017, with a collaboration with my good friend Pandelis Georgiou in Imperial College, we have designed a potential stat, just the design, I'm honest, never fabricated, but on simulation, it was possible to have a 10 by 10 potential stat, apparently, with CMOS 018, quite nowadays old CMOS technology. More recently, another fruitful and lucky collaboration for me, this time Paolo Crovetti, Politecnico di Torino, is a very bright guy in electronics. He already kind of 5-10 years ago decided to play in digital by replicating in digital the typical function of an operational amplifier. So you see few transistors, some logic gate, and then with the typically time domain working, he succeeded in demonstrating the possibility to design an operational amplifier. So we started a collaboration and we designed a potential stat. So the electronics for driving an electrochemical sensor. And in this case, layout, post-layout simulation and real layout, 23rd by 20 micron, including I/O stages. This guy has been produced, is now under test in our lab. What's about the data link? Well, first trial was not so successful. In fact, it landed only in an archive, never published. The problem was still too big. And the reason was that the ring oscillator, the starved ring oscillator, consumed close to 60% of the area. And this is typically done by due to the fact that we have, capacitor that need to support the frequency. And capacitor, in CMOS design are typically, when required, enough area angry. So we had an idea to change technology. This is 018 micron CMOS technology, UMC. We move it to 28 nanometers, and we discover we can succeed in designing capacitor-less ring oscillator because we can exploit leakages. We can exploit, better the parasitic capacitors. In this case, we did it 5 by 10. Again, only the design. Then, of course, we got an idea. We went back to 018, and this is fabricated and tested. We are going to present in Sofia this June. You see, 10 by 5 again, but done in 018, again, by realizing a ring oscillator without capacitor, so capacitor-less. So it seems that we are not exactly at 10 by 10, but you are close to 10 by 10 also for the main angry area consuming component of our backend for the transmission. What's about powering? About powering, I already mentioned you the work done in Berkeley. They worked with ultrasound powering. They did it. They also demonstrated in Vivo a few years ago. You see 2020, Nature Biomedical Engineering. But it's 1.7 millimeter cube, the total chip. And if you want to scale down by 30 by 30, the problem is how to integrate the piezo in your tiny chip. So we thought to still staying with the inductive link. Good friend of Gert and me in John Hopkins, Ralph Etienne Cummings, did it in 200 micron coil for application to brain. So we started working from this from the work of Ralph, and we imagined 3 scenarios of our body data. So, for example, for translational medicine, for in vitro diagnostic, we can mix whatever sample with our diagnostic and then put it on a plate, or in body humans, but close to the surface. So one of my students thought, for example, if we are in the heart, we are enough close to the external part of the body, so not very deep. And then we have computed here the expected size of the coil, and we measured for 150 to 65 microns. So not yet 10 by 10, but we are going below 100 or below 200. These are some chips that we have designed by following this funny idea of the body dust, and in particular, this one is more promising. It's 150 by 150. You see here the coil. The coil now is done fully by exploiting the CMOS technology. So these are several metal layers inside the chip that are providing an inductor that is supposed to receive the power. And you see here inside the other part of the CMOS that implement, for example, our potential stat, or in another version of the chip, we plan for neural stimulation. These are preliminary data that we have started getting on the coil, where it seems that we are capable to receive enough power. I didn't mention, but the potential stat I have designed with Paolo Crovetti from Torino is asking 9 nanowatts, so very, very negligible amount of power with respect to what usually we are doing in inductive link. Here we are shrinking down the size, we are shrinking down as well the PoE, so the received power. By the way, it seems compatible with the amount of power we need by the potential stat. And then, by the way, especially for brain application, we have succeeded recently in getting 2 orders of magnitude of improvement in the received power by moving from a passive resonance intermediate coil to an active one. So we receive the power, we have an IC that change the frequency of the received signal and then retransmit, and we have another series here of smaller resonant coil in order to succeed in powering our chip. And with this idea, we succeed in receiving enough power to a baby that you see is very small. This is neural dust in the sense that is micron size pixel with stimulating electrodes, with the idea to distribute thousands of this chip in the visual cortex, which is right here, in order to return vision to totally blind patients where there is nothing to do in the retina or in the visual nerves. Well, as we have seen in the very challenging list of things to do, it seems that the first 3 are close to arrive. I have showed you solution in 0.18, so we have not yet seriously tried to move down to 28. We've done once to 22 or to 14, where we will save for sure a lot of space. It seems we are coming with the powering. I had no doubt to go through the detail. I apologize, but if you're interested, I'm happy to share. We are also to exploit the concept of biofuel cell to see if we can receive local power, for example, by local content of glucose. And then, of course, we are still much away from any application, at least in animal. But, you know, life is long, never give up. We will see in 10, 20 years from now. Okay, so I hope to have convinced you that if we want to bring us by this time cover the human never offline directly from the skin, we still need, you know, a lot of dedicated electronics because we need to make smaller possible with a lot of function on board. We definitely need help by the biotech because I didn't mention, but it's easy to buy glucose oxidase on the market, enough robust and enough cheapest. But if you start just measuring lactate, it's another story. So we really need help from the biotech field in order to have all the biological material we might need at a decent price. Nanotechnology will help to improve the performance of our sensor. And if we integrate all of that, we can easily develop portable, wearable, and implantable, and might be one day also kind of spreadable diagnostic and kind of body dust. Of course, I have an enormous debt of gratitude to all the folks that work with me in EPFL, work now and worked in past, and I thanks them and thank you all for having listened my talk. Thank you so much.
Questions?
If you can't get the dust down to 10 microns or whatever, wouldn't that affect the fraction that got absorbed as opposed to like none getting absorbed? So is it a hard cutoff? Like if you can get it down to 1.5 times what you want it to be, when some of it still get absorbed.
Okay, so no idea. No idea, because I'm mimicking an answer I listened in Apollo 13. This is the problem 3575. We are still asking ourselves how to solve the number 3. So that's true. This is one of the many questions. I mentioned to exploit, for example, the internalization, we suspect that we need also flexibility. Well, it has been demonstrated that CMOS electronics below 30 micron of total thickness, and we have technologies to go down. I showed you the neural dust, with the stimulation electrode, we thin it down to 30 micron. It has been demonstrated by colleagues that if you reach 30 micron, you can be flexible with a silicon chip. So there is the hope to be flexible enough. As I showed you, even for bigger chip is not a big problem. The biocompatibility, I have no idea how it will be the internalization and externalization biological process. So we need to have this powder to make a trial. I have a friend in the Institute of Biomedical Engineering in Bellinzona, in the Italian side of Switzerland, that would be more than happy to work on that, but we still don't have the answer because it's a faraway problem that we will afford sooner or later, but we have not yet the answer. Sorry about that.
Yeah.
I want to something to the question is great talk.
Thank you.
I think in order to be internalized orally, we actually need to be about like 500 kilodalton, and that's like about 5 nanometer precise, because otherwise, things will not pass through the mucosal tissue in the GI tract or the nasal cavity.
Well, I have to be honest, I don't know, because I have already listened about this. I have biologists and medical doctor that told me that in principle, internalization is working also with white and red cells. And I have read literature of the fact that passing from the bloodstream to the biliary ways is done also by bacteria that are 7 micron. So if I have understood correctly, there is even not necessarily consensus in the medical field about how might be the desired size. Different.
I would never work with doctor on that. I wouldn't know, right? I think that drug delivery folks are people because they work on formulation with nanoparticles.
But that's the point. I agree with you. If when I read article in drug delivery, I found that numbers. But when I read article on the biliary system, how it works, I have found experiments with data that show that a bacteria by 7 micron was capable to pass through. So I'm not an expert, so I cannot tell you what is the reason why that bacterial succeed in passing through the barrier, but this is what some expert told me. If you can stay around 10 micron, you might have the probability to pass through.
Yeah, but we can have a discussion. The bacteria internalization have the different pathway.
I know.
But having said that, it's
But I have to be.
There's already an application for what you're developing for GI tract monitoring, right? So because it's much easier, they can sit there for a while, and then you can just send, I don't know, a magnet to retrieve them, so they don't even have to stay there, or the mucosal tissue constantly gets degraded and it washes off things. And I think it's a huge application there for like Crohn's disease or IBD disease, as they want to continuously monitor this. So I know, generating nano-sized electronic might be really the future, and we probably have to go to 2D material and things like that. But there is already application for. Have you considered those?
Yeah, absolutely yes. Especially to answer reviewers that didn't want to fund the research. Yes, one other application might be, for example, colorectal cancer. So in that case, you don't need to pass through the internalization. You do everything in the digestion systems. And then once you are in the region, it is supposed the particle attached to the cancer cells. If there is no cancer, it is supposed they will go till the end. So I totally agree with you. I usually refer to the internalization because it set the challenge. I need to stay smaller possible, because for tracking this, I can even do probably with 100 micron, with 150 micron, because there are already technology in the market when I can swallow a camera that is a bit bigger like that, and it is recovered after passing through. So yes, from the standpoint of selling the technology is a very good point. For the standpoint of a bad boy that look to find science fiction, is much way more challenging to target the problem of the internalization, even though I agree with you, we are still a lack of, biological information in that direction. And the B plan eventually would be to track the colorectal.
Yeah, so in addition to like enzyme-based electrochemistry, are you considering other mode of detection, electron tunneling?
Well, in principle, I'm using whatever kind of detection. So here I've just presented, the selective membrane or the enzyme, but we have branch of research in the lab that, for example, work with the memristive nanowires. So we have silicon nanowires that are in between 30 and 50 micron of width, where we have discovered, memristive effect or whatever kind of switching effect. And we have succeeded in inventing a detection method. Actually, we have in the room the lady that did it first.
But they can get detection aid because you'll be limited by whatever enzyme you can have access to. In theory, like metabolite can be detected by even optical method, like some other kind of electrical method, like.
Yeah, but I see two issues in this discussion. So the first one is the selectivity. So yes, I can detect by optical method, but then I have to be sure that I'm detecting the glucose and not the lactate. I have just closed a few months ago a PhD thesis where we try to use Raman spectroscopy to measure on the skin glucose, lactate, and urea, and we have found tons of interferences. So whatever other molecule that change my signal, meanwhile, I'm trying to measure the glucose. So here, if I have the glucose, for example, oxidized on board, I'm sure that if I'm receiving a current, it's only the glucose. The second issue that I see is the miniaturization, because again, as you have seen, I've already done a potential stat, an inductive link in 150 micron. At this moment, optics promise one day those scale, but they haven't yet shown. So the miniaturization and the selectivity problem, in my opinion, have to be considered.
No, so if handling these implants are such a problem, either with ultrasound or just wireless nowadays, why not consider self-power? Why not considering enzymatic batteries, these bio batteries, or supercapacitors, even actuators with vibration? Because your devices, your chips seem to be something like nanowatts. That's really nothing.
Yeah, I totally agree. Thank you for the question. The answer is definitely yes. So we are trying to develop a collaboration with an expert who is in Dresden on the kind of micron batteries based on kind of supercapacitor based on electrochemical fuel. We are also trying to integrate biofuel cell. We have just done our first research last semester. The point is that when we started 15 years ago working on implantable, that time, the state of the art of all the research we have seen in the literature were playing much more in terms of robustness and the quality of the received energy, were playing in favor of the inductive link. So that's the reason so far we still are keep going with the inductive link because we are still, let's say, receiving help by many research and real products that are based on the inductive link. So what you mentioned, yes, but it's still more on the research side. So even the people that are developing this kind of supercapacitor are still looking for applications. And I agree with you, our might be one of their good applications, but this we have just started now. So I have no data, I have no sure answer to provide you. But I agree with you, there is a lot of promise there.
An inductive link can be used to charge a supercapacitor anyway.
Exactly, exactly. But this, what we are discussing is to use, for example, a supercapacitor, which is not exactly supercapacitor, is a micro battery based on idea like supercapacitor that is directly providing energy.
Powered by the metabolism too. Yes.
Yeah, exactly. Like in case of biofuels.
The last question, yes.
Very interesting talk.
Thank you.
My question is, in the future, that what do you think about the compare between the CMOS with the organic technology?
Another very good question. So, you want a polite answer or you want my personal opinion?
Go for it. Record it.
Unfortunately, we are recorded. So my personal opinion is that no way. So imagine what is the level of investment worldwide by industry and companies in CMOS technology, and then compare with investment in organic electronics. So I'm not saying that organic electronic is not useful. It's useful, absolutely, but for developing some special devices and cases. So so far, we have seen even some architecture done by polymeric transistor, but I just highlighted they are always quite simple architecture, useful but simple application architecture. That might be extremely useful to be in future introduced in, I don't know, an RFID sensor for solving some dedicated problem. But if we speak about electronics, that means, tens, 100, 1,000, millions, billions, trillions transistor in some centimeter square, no way to compare in terms of robustness, in terms of state-of-the-art technology. So for probably the next 50 years, humans.
50?
Second? Yeah, 50, in my opinion.
It's recorded. We'll go back in 50 years.
Okay. Sorry about that.
Thanks so much.
Thank you so much for being with us. Gert, close recording.
Yes, now I can just, yes, that is, yep. Oops, still recording. Still recording, yeah, that's fine.